So hello and welcome to the Xenopus laevis basics of development, and I'm just giving you a little overview about the basic happenings of the development and of the molecular mechanisms. And I want to start with showing you this little circle here, which should be the oocyte and also the zygote. So I just made it without the colors because these colors should be laid on the germ layers, and because we don't have really germ layers on the oocyte and zygote, that's why I just left the colors. Alright, so let's start. I'm just naming that here the oocytes, or the zygote later on. And the important thing here is to know that we have actually also like something like this here, two hemispheres, in which this hemisphere is called the animal pole and this hemisphere is called the vegetal pole. This side here, well, I'll have to tell you something else first. This here, let's say, is the sperm entry point. All right, it can be anywhere on this hemisphere, so it can be here, here, here. It just has to be on the animal pole. And let's say the sperm entry point is here, then the opposite point here, a little bit upwards, will be the dorsal side. So here we have D, and that's the ventral side of the blastula, and later on also of the embryo. But you know, there are a few shape changes. That's why the dorsal side later on will be about the dorsal side, but not exactly the dorsal side of the finished amphibian frog. So now it's important to know that this here is the sperm entry point. That means that in the oocyte, you know, you have now two nuclei. That means one nucleus from here and one nucleus of the whole cell. That's one cell here at the beginning. So you don't have this typical hemisphere thing firstly, but it will come. Then you have the zygote. And what happened is that the nuclei fuse and you have one nucleus afterwards. And let's say it is here somewhere. And afterwards you have some cytoskeletal rearrangements. That means that the first thing that will happen is some microtubuli, let's take another color, will form somewhat like this here on this side. You know, there are also microtubuli here somewhere and whatever, but these are important here because we have the minus end here and the plus end here. So the plus end is where it polymerizes quicker than it depolymerizes, but that's not so important now. What's important is that you have multiproteins. Well, you have kinesin, and you also have guanine. And kinesin is the important one here because it goes in this direction, from minus to plus. That means that all proteins that are somehow connected to this multiprotein will be traveling from here to this direction and will be then later on here. And the important proteins I'm talking about now, which are connected to kinesin in this zygote, are, well, let me draw them here with another color. And the one, they are disheveled. That's disheveled, and also GBP, right? So let me write that here. So DSH stands for disheveled, and GBP stands for GSK binding protein. So it binds the GSK, and GSK stands for glycogen synthase kinase. And these two are now in this hemisphere. And there's another one that is also in this area here, which is X-Wnt11. So these three are important. And now you might wonder why is that so important. And the reason is because these three proteins are part of a pathway which, I'll just quickly try to draw it here. You have the cell here, you have a receptor which is called frizzled, and you have Wnt. And Wnt, if Wnt binds to this receptor frizzled here, frizzled can bind to GSK3, and GSK3 is, the glycogen synthase kinase, and now this is bound to frizzled, and it can't do its work, and its work is to phosphorylate, which is, a kinase is usually phosphorylating other proteins, and this one, especially here, would usually phosphorylate beta-catenin. And that is the important thing here. So beta-catenin is here the most important thing, because now that Wnt is here, that GSK3 can or is bound to frizzled, to this receptor here, to the intracellular domain here, somewhere here, of the frizzled receptor, and beta-catenin won't be degraded because it won't be phosphorylated, because that can't happen here. It's not phosphorylated, so it won't be degraded, and it can go into the nucleus. And disheveled is also part of this pathway. It's important for binding that GSK, and a glycogen synthase kinase binding protein is important for binding it, and therefore also important for preventing that GSK can do its work. So that are the basics, and now you have something you have here. So the whole process here results in beta-catenin being here, right? And I'm going to show you that again. Wait, one thing that is still important as well is that you would think that all these proteins are just in here, but they're also a little bit more upwards. And the reason is because the cortex, which is this area around here, will rotate relative to the cytoplasm about 30 degrees in this direction. And therefore all the proteins that are firstly here, a little bit more in this direction, right? Okay, let's go on. So I'm showing you the gradients here. So you have beta-catenin, which is, around here somewhere. So on the sorry, that's the dorsal side. On the dorsal side here, right? And that's the ventral. And at the same time, you have another protein, which is VegT, and also that one, VJ1, is also important. And these are just in this hemisphere here, so in the vegetal hemisphere, around here. So these are factors that are really important for endodermal cells, so that what later will be endoderm, which is yellow here. So they are only here. Beta-catenin is here, and then what's also important is that beta-catenin and VegT are both transcribing something, or important for transcription and translation of something that is called X and R. So X, we had X already here as well, here with X-Wnt. X always stands for Xenopus, nodal related. That just means that nodal is a factor, so that one is a factor that is important for a specific signaling pathway, and so it's just related to this protein, and it comes, or it's a Xenopus protein. And this one is, made if these are active. So here it's the most, obviously. So here you have a lot of it. And here you will have less. And that's really important later on for determining which cells here are which kind of mesoderm. All right, I just tell you something else now, which is BMP. And BMP is the bone morphogenic protein. Bone morphogenic protein, which is part of the TGF-beta signaling pathway. So you just have to learn this pathway of TGF-beta to know what the bone morphogenic protein does. So it basically is important for SMUD proteins that are important for signaling. And BMP is usually all around the cell, so here on this side. And where it's not is in this area here, because here it is inhibited by something, right? And then, well, I'll show you that here. You have also something else, which is what you will find here. That is X-Went8. We had X-Went11 here, and that is X-Went8. So that's really important to differentiate, because X-Went11 is in this area here, and X-Went8 is just here in this, in this area, which will be the mesoderm. And this is also called the equatorial region, or the marginal zone. So write that down here. Marginal zone. Right. Now I'm going to show you some details. Before I'll explain you why you all need these factors and proteins and why they're so important. So we talked about the sperm entry point. We talked about how the dorsal ventral axis is determined. Now we have cleavages. Cleavages are some kind of mitosis, but without cell growth between that. So you just have cleavages, and the whole volume stays the same. So the volume of these cells, of these two, are the same as these 8 here, for example. Yeah, and these are the polar cells, which come from meiosis. So you just have to understand what meiosis is in order to know what the polar cells or polar bodies are. And you have cleavages, and these cleavages have different axes. So you have meridional cleavages. This one is meridional, and this one as well. And this one is equatorial, right? So these are on the poles here at right angles to each other, and the third one is equatorial, but a little bit pushed upwards. And this is, it's an easy reason, because it's because of the cells here. The cells here are yolky cells, so they have a lot of yolk. And yolky cells, or yolk in general, interferes with cleavage. That means also here, for example, you have a fast cleavage here, and here it gets slower, and here it gets slower as well, and here as well, because on the Vegetal-Pole, so here we have a lot of yolk, and yolk interferes with cleavage. And here it interferes in a way that it says, okay, it pushes upwards the line here. And where you can also see that pretty good is if you have a look at other organisms like zebrafish or chicken. If you have a look at these organisms, you will see that the embryos don't have any cleavage here. And that's because they have way more yolk than these cells have. So they also interfere with the whole process. And such a cleavage mechanism is called holoblastic. And there are also other cleavages, or there are other forms of cleavages called, for example, meroblastic. Meroblastic is a cleavage form in which only the upper part is cleaved and the part down here, so the Vegetal-Pole, is not cleaved. So holoblastic is, holoblastic, we have Xenopus, and meroblastic, we have, for example, Danierario, which is the zebrafish, and another one would be chicken, so Carlos. They have meroblastic cleavage. Alright. Another thing is, the first cleavage here takes place after 90 minutes of fertilization, and all the other ones in an interval of 20 minutes. Alright, so these cleavages take place now until something forms. So from the 16 to the 64th cell stage, you have something that's called morula, because it just looks like a morula. And then afterwards you have a name that is called blastula. So blastula is just a typical stage in which something like this forms. It has way more cells, I'll show you soon, and a blastocoel forms, but we'll come back to that. Firstly, there are cell stages between. For example, the 12th cell stage, or the 12th cleavage, produces, I think, about 4,096 cells, yeah. I mean, you can calculate that on your own, but that is really important because something happens here. That's the Midblastula-Transition. The Midblastula-Transition is so important because what happens here is that this is just the point, Midblastula. It's just the middle of the blastula phase, which here is, I mean, it's funny, but it's on it's the late blastula stage here. But Midblastula is just found, so this stage here is just found on another organism, and that's why it's called like that. But in this organism, it's just on the late blastula. All right, and what's important here is that the zygote, or that you have now zygotic genes turned on. Zygotic genes are turned on. Before that, you would only have maternal genes, so genes that were already, or maternal proteins actually, that were inside the oocyte and still are in the oocyte. But now, or are in the embryo now. But now, the whole embryo starts to produce its own proteins, and the maternal genes just lose their importance. Now, having a look at this here, this shows how the blastula looks like. And I like this picture because, I mean, the colors are not so good because usually, like I showed you before, the colors of the main books, or of the best books, I guess, on these topics are always yellow for Endoderm, red for Mesoderm, and blue for Ectoderm. That's just different colors, but imagine that being red, and that blue, and that yellow. And you just see perfectly how the Mesoderm is like a belt around here. And what's not shown is that the Endoderm is usually covered, or is covering, the Mesoderm here. So that would be blue as well, and just inside it would be the Mesoderm. So usually this blue here would go over that here. So the Endoderm is covering the Mesoderm here. But that's just good for visualization. All right, so, and that is the blastocoel here. And yeah, let's go back to my drawing. So why is it so important to have all these factors now? I will show you why. Because if we now imagine that being the blastula, we have the ectoderm, the mesoderm, and the endoderm. And the endoderm is usually the endoderm because it has factors like VegT or VG1 or something like this. And the ectoderm has also its factors that say, well, okay, I'm an ectoderm. But the mesoderm is induced by cells, or will be induced by cells that are, for them, induced by the endoderm. So the endoderm induces these cells here, the mesoderm, to be mesoderm. That was done, or that was shown by an experiment where they took such a blastula and just cut out here the mesoderm. So, getting back to this picture, they cut out this green area here, and that then put this animal pole onto the vegetal pole, so the ectoderm on the endoderm. And what happened was the ectoderm got the mesoderm, and the ectoderm usually never gets mesoderm. So the reason is, if the animal cap here, or if the animal cells make contact with the endoderm, which happens here, for example, then they form mesoderm. And that is one of the reasons why the blastocoel is so important, because it prevents the ectoderm from connecting to the endoderm. And therefore, they would make mesoderm otherwise. So, and what are the factors now that are so important for this mesoderm? Well, having a look at something that is called the fate map. The fate map shows us what will be, or which cells are going to be what. And we know that the ectoderm here will be the so-called epidermis, right? These cells will be the neural cells that form the neural tube later on. And the mesoderm, we have the notochord here, and also the organizer region. Here we have the somites, and here we have, I guess, blood, and the kidney should be here. Alright, and the heart, I forgot the heart is also here. Alright, so that would be a fate map. So please don't confuse it with a specification map. A specification map is if you take these cells here in the Blastula stage and look and somehow cultivate them, and then look what they're going to be, or what they make at this stage. And if you take these cells out, they won't form somites here. They will probably form mesenchyme or blood or something. So if you take these cells out, they will form something else. And that is a specification map. And the fate map is if you let them inside the blastula and have a look what it does later on. So it's affected by a lot of different signaling pathways. It's affected by different kind of cells around here, of the epidermis and of endodermis and all these areas. So don't confuse fate map and specification map. And this is the fate map. Specification map would just have epidermis around here. So it won't have any neural cells here. And I'll tell you the reason why later. First, the specification map would also have mesenchyme here and blood all around here. And the somites, well, they would be, I guess, around here somewhere, and the notochord as well. So that would change a little bit. And the reason is, well, we already talked about what the endoderm will do. It's here, and it has these two factors. And the ectoderm is usually going to make neural cells. So the default state of ectoderm, so default state here is neural cells. And we talked about PMP already. So that's here. At the beginning, PMP is all around here, and it's inhibited here. So, if I have a look at it again, PMP is, for example, here, all around here, we have PMP. And we don't have PMP here, because it's inhibited here. And that's the reason why BMP, well, why this area here is epidermis, because BMP plus ectoderm is epidermis. And epidermis without BMP is, default state, which is the neural cells. And then you have some factors here that are inhibiting, but I will come back to that soon. Firstly, I want to tell you about the mesoderm. The mesoderm is something that is induced by a lot of different things. For example, by signal 1, which comes from up here, which is VegT and VJ1. And then we talked about beta-catenin, which is around here. And beta-catenin plus VegT are XNR here. So XNR is a lot here and makes a signal here. So signal 1, signal 2. And signal 2 is actually something that is called the Nieuwkoop-Center, which is the dorsal side of the endoderm, and induces here the organizer in this area. And the organizer is basically just a third signal, which is an inhibiting signal here, here, and here. And the inhibiting signal is for the fourth signal. The fourth signal is BMP, which is also here around, and Xwent 8, which I showed you before, is just here in this area. So it inhibits all these things. And the inhibiting factors are noggin, chordin, and Cerberus. So for BNP, it's noggin, chordin, and Cerberus. And for X-Men, it's also Cerberus, and I guess something that's called frisbee. These are just molecules that bind to, for example, BMP or X-Min8, and so they neutralize them. So X-Min8 and BMP are the fourth signal, so right here, the fourth signal. Then you would have the third signal, which are these inhibiting signals that we have here, so these ones. Then you would have the second signal here, and then you would have the first signal, which is here. And all these different signals are therefore marking this one and this one and this one. And an experiment that was also done is, remember the experiment that I told you before, is cutting here the mesoderm out and putting the animal cap between these endodermal cells. So you have like a cell, and you have some cells here, which are the here the ectoderm, and well, somewhat like this, and these cells around here are endoderm. And what will happen is, it depends on where I take this endoderm from. If I take it from here, for example, then it's, the area that forms here, this signal here, that forms the Nieuwkoop-Center Organizer. So we'll have also Nieuwkoop-Center Organizer out of the ectoderm, because the endoderm induces the ectoderm to form something, and it's the endoderm from here, so it forms Nieuwkoop-Center Organizer. If I put it from here, or if I take here the endoderm and use it here, then it will form, plotting kidney and somates, and so yeah. Another thing that's really important is that these area, or this area here, is the dorsal area. That's the ventral area, and that is more a lateral area. So blood and kidney are more lateral. So one thing that is really important is that this area here, which is induced by the Nieuwkoop-Center, is the so-called organizer region, and it is the Spemann-Mangold-Organizer, so SMO. This is this region here, or more accurately, it's just the cells around here, which will later form the blastopore lip, and the blastopore lip is part of the blastoporus, which will also be here somewhere, and which is part of the gastrulation process, which I will talk to you, or which I will explain to you right now in a video. What I'm going to show you now is the gastrulation, and I have a cool video here that I will show you, or that I will post you down in the description, which I think shows the whole process pretty good. And the first thing which you won't see if you have a look at the embryo, because it happens inside, is that this form of endoderm, which is in the blastocele floor, because that is the blastocele, that is the ectoderm, that is the mesoderm, and here's the endoderm. And the first thing that happens is that a blastocele floor on the dorsal side will move up, migrate upwards. So the cells here migrate in this direction until they are above here, above the mesoderm here, which is the red thing here. And only now you will see a little groove here, this little groove, and this is actually like a slit. I'll show you later how that looks, or maybe I have it here. So, well, it's not really seeable, but you see it on the second picture here. You see here, this is like a slit here. That's what's here, so a little slit, if you look at it on the off the dorsal side here. And this here are the so-called bottle cells. These are cells that are here on the outside usually and migrate inwards, but want to stay here. So they're fixed on this side, but migrate inwards, and that's why they have such a look here. So this long look. So they are a little bit slender-necked. And they are important because they somehow initiate the involution. So it's no invagination. It's important to understand the difference between invagination and involution. Invagination is something where you would have, like here, such a form around here, and involution is something where something folds in here. And if we have a look at the whole process, you see it folds in around here. Right. So you have the bottle cells, and these bottle cells here migrate inwards, and afterwards, after they did this little groove here, they're not needed anymore. So you can't put them away now because they somehow initiated this process. And now cells from the inside have contact with the cells from the outside, and the whole process already just goes away. So the whole process now is somehow started, and now you don't need these cells anymore. They're just important for initiation. And now this involution process takes place. So the mesoderm here, the mesodermal cells, plus the endodermal cells, which I'll tell you later what they exactly are, but they will undergo something that is called convergent extension. So this here will converge, it will get smaller and extend in this direction. So cells migrate here, and if a lot of cells migrate here, so if this here gets smaller, then this gets longer. So there is no mitosis or something like this. So there's just a cell migration process. And here on the top you have endoderm. And this endoderm is the so-called prospective pharyngeal endoderm. So endoderm that is really later on the anterior. Below that you have here mesoderm, which is the pre-caudal plate. So before the cauda, pre-cauda, pre is before, and cauda is the notochord. And yeah, another name is the prospective head mesoderm. And here you have, or later you have the cauda, the cauda mesoderm, which is this here, cauda mesoderm. And these cells here on the front, so the prospective pharyngeal endoderm, if you take this plus this here, which is the precaudal plate, you can also call them the head endomesoderm. So this here. And they're called the head endomesoderm because they will form the anterior parts of the embryo. So these cells here will migrate to the anterior part. So they move forward. And another thing that you'll see is that here, so here you already see this slit here. This is the future achenteron, which is the new coelom, so new hole that it forms, the gut, you know. And this is the blastocele, which gets smaller here. And the blastocele, you wonder, why would I need a blastocele if I form a new hole here anyways? And the reason is because here you want to prevent the endoderm from connecting to the ectoderm. Because, like we talked about before, if endoderm is connecting to the ectoderm, mesoderm will form. And you don't want mesoderm here. So that's an experiment as well. You take endoderm and you surround it, or you surround the ectoderm with endoderm, and out of the ectoderm, mesoderm will form. So that's why you need a space between them. And another theory why you would need a blastocele is because you need space inside the whole thing in order to be able to make another hole inside it. So physical reasons. Yeah, so these are the two reasons. So this here is called convergent extension here, and it is a form of mediolateral intercalation, which already tells you what it is. Lateral parts on each sides are going to medial parts in the middle, and then you have an intercalation, and they just migrate in the other direction. And at the same time you have here the ectoderm around it, which will migrate. So if we go back, then this is here the animal pole, and now cells from down here, So from down here, it will migrate upwards in this direction, and then this one here gets smaller, but this and that gets longer. So it can surround the whole embryo, which is called epibole. So here, now the ectoderm surrounds the whole embryo here. We'll see that here. And now, obviously, the animal pole changes its position. So we had it before, it was here, the animal pole, and now it migrates. We'll see it here, around it here, here. So the whole directions also change. And what you see at the end here, that's the dorsal lip. The blastopore was here initially, so it was, on this direction, but it's now here because, epibole forms. And this is the dorsal lip, and now also other lips around it form. That's the yolk plug. So that's the endoderm that's still here. So a yolk plug, and here the lips, and the dorsal lip is usually the organizer. Yeah, here you have the slit-like blastopore, and down here you have the yolk plug, which you see here. That's this here in 3D. And then you have a dorsal lip and a new lip here again, which is called the ventral lip, and the lateral lips. Yeah, and then afterwards you would have, so what I'm not going to discuss here again is the neurulation, but that would happen afterwards. So afterwards you have, different forms of ectoderm here, the neural ectoderm and the epidermis. And then the embryo would flatten here, so, it actually shows a little bit of the later stages. Let me see if I can stop it. 4, 5. So it will flatten a little bit. And then here, you would have the neurulation on this side. And here you have the mesoderm, which is on the dorsal side. So this is not a dorsal side, this is the ventral side. Yeah, and down you have, anterior, posterior, and the mesoderm here, and here the head mesoderm. The mesoderm here forms the notochord, and above it you have the neural ectoderm, which will form the neural tube by here, firstly flattening, and then there's also kind of involution process, so it's something different. And then the epidermis grows around the embryo here. So, yeah.